and welcome to this lecture titled Introduction to Microbiology. My name is Mohammed Alkharraz. In this lecture, we're going to talk about the different types of microorganisms. We're going to also talk about normal flora. We will talk about the role of the microbiology lab in aiding in the diagnoses of patients. And finally, we're going to talk about why do we study microbiology. So as an introduction, we're talking about microorganisms. So what are microorganisms? Microorganisms are very small creatures which live everywhere in nature. And they're associated with the ecosystem in many different ways. But what we're important in is how do they interact with humans, and more importantly, how do they interact with human diseases. So they're classically divided into prokaryotes and eukaryotes. Prokaryotes have very basic cellular structure, prokaryotes are the same as bacteria. So they have very basic cells. They do not have a defined nucleus. So that means that they have very simple structures. However, eukaryotes are more complex. They have a lot of different cell organelles, such as mitochondria. They have different roles in terms of human diseases. So viruses, they use the host's machinery to replicate. Viruses do not have the ability to replicate by themselves. That's why they have to enter other cellular organisms, such as human cells or even bacterial cells, and they use their machinery to replicate their DNA or RNA. Some are associated with mild diseases, while others can cause some serious human diseases. An example is then the human immunodeficiency virus, HIV, which causes the acquired immunodeficiency syndrome, AIDS. We can treat viral diseases using different antiviral agents which tackle their replication process. Bacteria are the simplest living organisms. They are unicellular, and they're also called prokaryotes. They have a very basic cellular structure. They are abundant in nature, which means that they are found in many different places. They can be found in soil, they can be found in volcanoes, and many different places. We often think of bacteria as bad creatures, but they actually serve many beneficial processes, such as in the fermentation of yogurt and the creation of lebanon, for example. However, some are associated with very severe and very serious human diseases. As we said, bacteria have very simple structures, and our goal of learning their structure is to learn how are they different from our human cells, to eventually find some pharmacological agents that can tackle the bacteria without interfering with the human cells. We're actually interested in the structure of bacteria to learn or to know how we can treat them. Let's take a couple of examples: this bacterial cell wall and the ribosomes. The bacterial cell wall is very important because we as humans do not have cell walls. And that means if we have a certain drug that can destroy the bacterial cell wall, it would not kill our own human cells. And that's actually true. We have the penicillins, which tackle the bacterial cell wall, and they do not cause problems to our cells. It also turns out that the bacterial ribosomes, first of all, the ribosomes are the cell organelles that are responsible of taking the messenger RNA that comes out of the DNA and forming polypeptides or proteins. It turns out that the bacteria have different ribosomes than humans, and we actually use this knowledge to develop different types of drugs that block bacterial ribosomes, such as the aminoglycosides, the tetracyclines, etc., macrolides, and all of these different pharmacological agents. When we talk about fungi, now we come into eukaryotic cells, which means that they have complex cellular organelles, and they're closer to human cells, which makes it a little bit difficult to treat and limits our targets in order to kill the fungi. But one of the very important targets that we use is the presence of ergosterol in their cell membranes. Ergosterol is not present in human cells, and we have, for example, the most famous antifungal drug is amphotericin B, which works by interfering with the ergosterol in the cell membrane. Fungi are some are round yeast-like forms, and other are filamentous branching forms. And they're not very famous of causing human diseases such as bacteria or viruses. However, it's most important in immunocompromised patients. And now we'll talk about parasites. Parasites are eukaryotic cells, and they vary in structure. Parasites have different types of organisms inside this group, but we like to just put them in one bag and call them parasites. What is interesting about parasites is that we learned some of their life cycles, and they have very complicated life cycles. So the idea behind this is that when knowing the life cycle of the parasites, for example, some parasites start from humans and they go to different animals and they come back to humans through sources. So in that way we can know which patients are at risk for getting this parasite, uh, by knowing their life cycle. These parasites are much more important in tropical areas, such as, for example, in Africa and Asia. And this raises a very important point, is that parasites are not very important in some areas of the world, such as in the United States, for example. And this poses a problem of drug development. Because when companies want to develop drugs or make researches and create new drugs, they eventually want to sell these drugs. And if the rich countries, such as the US, do not have any patients who suffer from parasitic diseases, no one in Africa, for example, can afford buying expensive drugs to treat their parasitic illnesses. And this just poses a small point that microbiology really has a lot to do with different aspects of life, such as drug development and poverty. Although we tend to think about bacteria as these evil organisms that try to kill us humans, they actually serve very important functions in maintaining our health. Normal flora are bacteria that are normally found in our bodies. And they're found in sites such as the skin, the gastrointestinal tract, the external ear, in our eyes to some extent. And these serve very important functions. But we should point out that there are some sites in our bodies that have absolutely no bacteria, and they are which are called sterile locations. And any presence of any sort of bacteria in these areas is considered abnormal or pathological. Some of these areas include the heart, the lungs, the blood, the central nervous system. So just to name some examples of how these normal flora help us humans in our lives, let's take the example of Clostridium difficile. So the gastrointestinal tract is full of bacteria, and the normal flora serve as they compete with the bad bacteria or the pathogenic bacteria, which are found in the gastrointestinal system. So, for example, we have this bacteria with this anaerobic bacteria called Clostridium difficile, which lives in our GI tract. But normally, when there's a lot of normal flora, there are a lot of bacteria, they can't grow and cause problems. However, when a patient takes antibiotics, for example, and the normal flora and normal bacteria die in the GI, and this Clostridium does not die from these antibiotics, it's kind of resistant. So the Clostridium difficile starts to multiply, start to increase in number, and then they start causing problems. They start releasing toxins in our GIT, and then the patients start having diarrhea. Also, the normal flora enhance the development of the immune system of the baby. Moreover, the normal flora help us very much in production of important nutrients and aid in the digestion and absorption of them. So, for example, a lot of bacteria in our GIT produce vitamin K, and this is why, for example, when a newborn is born, it is typical to give them intramuscular injections of vitamin K because they do not have a lot of bacteria in their gastrointestinal tract, and that puts them at risk of deficiency, vitamin K deficiency. If a child was to be born in a place where there's no good medical attention and they did not receive this vitamin K injection, it's very probable that they might have a condition called hemorrhagic disease of newborn, because vitamin K's normal function is to help in the clotting of the blood, and in case the child does not get the vitamin K injection and without the presence of the normal flora and the GIT, they might have this condition where they bleed. Knowing where bacteria are normally present can act as a needle of infection when barriers are breached. So, for example, when a surgeon wants to perform a gastrointestinal surgery or surgery involving cutting the GIT, it's much more likely that there would be a skin infection because the GIT is full of normal flora, full of bacteria, which can easily go out from the wound and cause an infection. Another aspect to know about normal flora is that we as doctors should know where are they present, and this guides us on how to deal with these sites, these locations, or these infections. So, for example, when performing procedures such as inserting a Foley catheter, which is a tube inserted through the urinary system into the bladder, we know that the urinary system, the urethra, and the bladder itself is a sterile place without normal flora. That's why when we insert a Foley catheter, we use a sterile technique where there's no skin contact with the patient. On the other hand, when, for example, inserting a nasogastric tube, the nose and the GIT is full of bacteria, full of normal flora. And in that case, we do not have to use a sterile technique. We just insert the tube, just wearing clean gloves. So when we learn microbiology, we learn a lot of different things, such as the structure of the bacteria and viruses and the organisms, the pathogenesis, the mechanism of transmission, the epidemiology, the relation of the organism with the host, and how can we detect them. And we just learn these things to learn how to treat patients, and that's the ultimate goal. So now we're going to talk about the microbiology lab. Microbiology lab is a very important part of microbiology, and it really helps a lot in the diagnosis and the management of patients. What we can do as physicians, we can take samples from the patient and send them to the microbiology lab. And the lab can do a range of different tests in order to help us diagnosing or even treating the patient. Sample, we're talking about the things that the patient has. For example, we can take a sample of the sputum of the patient. We can take a sample of the urine of the patient. We can take a sample of the blood of the patient. We talk about tests such as the gram stain, for example, which is a simple stain that is put on the sample, and we can see if there are bacteria present or not. The gram stain, by the way, stains the cell wall of the bacteria. So cultures that we take this sample, let's say for example blood of the patient, and we can put it into a growth plate. And this growth plate provides all the nutrients that this bacteria needs in order to grow. And then after the bacteria grows, we can first of all know that the bacteria is present. So if I put the patient's blood, for example, on a growth plate and I see bacteria growing, first of all I can tell the doctor, hey, there is bacteria here because I can see them growing in front of my eyes. And secondly, we can know which antibiotics work best on this bacteria. And we do that by putting like drops of antibiotic on the plate. And if it kills the bacteria, we can tell the physician, hey, this antibiotic works. If it does not work, can tell them, be careful, this antibiotic does not work. And this really helps in the management of patients. Also, we can do a number of different more fancy tests, which, for example, we can look for the DNA and RNA of the actual organism. Most commonly is used for viruses, but it can also be used for bacteria. But these tests are a little bit more expensive, and they're not typically or routinely done.